Hello again. When we last met, we were talking about simple stains, and you'll recall that with our methylene blue dye and our Congo red stain, we were coloring everything the exact same color. So it didn't matter whether we were looking at yeast cells, that is Saccharomyces cerevisiae, or if we were looking at bacterial cells such as E. coli. No matter the cell structure, everything stained the same color. There was no differentiation between colors. That's why we're here today to talk about differential staining, where the color actually matters. That is, depending upon the nature of the cell, we're going to see that one type of cell stains one color, another type of cell stains another color. And that's why I've donned my tie-dye lab coat today for this lecture. I thought it might be cool to exemplify differentiation based upon color. So we'll talk about what it means in certain stains, such as the Gram stain, if something stains pink or if something stains purple, and look at that, what it indicates about cellular structure. Today we're going to cover the Gram stain, the most famous of all differential stains, and one that will be a part of your life from here on out if microbiology stays a part of your life, as it should. And we'll talk about the acid-fast stain as well. So let's begin with the Gram stain, developed by a Danish physician in 1884. He was an incredibly humble man, and he was working in a hospital morgue in Berlin, actually, where he was working at trying to differentiate the post-mortem bacterial populations in victims of lung disease. And he was able to find out that he had a staining technique that allowed some bacteria to retain the initial stain while others lost that initial stain. And so he called those bacteria that retained the initial stain, he called them Gram positive, whereas those that lost it were called Gram negative. So this is where the terms Gram positive and Gram negative derive. It has to do with those cells that retain the dye called crystal violet. We call them Gram positive. Those that lose that crystal violet dye are called Gram negative. So don't confuse these terms positive and negative with any sort of quality of the cell besides its ability to retain the primary stain. But let's look at what actually was elucidated to be the cellular structure that allowed some cells to hold on to the dye while others couldn't. So those that could retain the dye were called Gram positive, and this is what the structure of a Gram positive cell looks like. Notice that its quintessential feature is a very thick layer of peptidoglycan. In fact, Gram positive cells may have as many as 30 sheets of peptidoglycan. This is a polysaccharide with a repeating structure of two sugar derivatives called N-acetylmuramic acid and N-acetylglucosamine, NAM and NAG for short. These chains repeat and form this very thick layer. Peptidoglycan has kind of a feature of a metal tea strainer. It's very porous, it allows things in and out, but it provides rigidity to a cell wall. So the rigidity of a Gram positive comes from that thick peptidoglycan layer. Also, characterizing the Gram positive cell wall are two types of acids, lipoteichoic and teichoic acid. Notice that they're differentiated by their attachment site. Lipoteichoic acid attaches to the cytoplasmic membrane. That makes sense, right? Because it attaches to lipid, lipo. Whereas teichoic acid attaches to NAM residues, to the sugar derivatives in the polysaccharide. Both teichoic and lipoteichoic acids are sometimes collectively called the teichoic acids. They are polymers of either glycerol and phosphate, or ribitol and phosphate. So alcohol, phosphate. And all of you remember that phosphate is an anion. Anion, negative charge. So this means that a Gram positive cell wall has a negative charge. And this is true of all cell walls. So the negative charge of a Gram positive cell is imparted by the lipoteichoic and teichoic acids. Now this structure is very different from that of a Gram negative cell. You can begin to think about why this cell structure might hold on to the crystal violet dye, whereas the next structure, a gram-negative cell, would lose that dye. One big difference that we note right away is the thickness of the peptidoglycan. In a gram-negative cell, the peptidoglycan is very thin. We also notice that the location of that peptidoglycan is different. So remember, in a gram-positive cell, the peptidoglycan was the most outer exposed layer, whereas in a gram-negative cell, it is protected inside of what is called the periplasmic space. Now, the periplasmic space of a gram-negative cell is teeming with action. There are enzymes involved in nutrient acquisition. There are proteins that are enzymes involved in building the peptidoglycan. There's a lot of workers in the periplasmic space, and sometimes that periplasmic space takes up a huge amount of the volume of the cell, 20 to 40%. Notice also that another protein inside of the periplasmic space plays a key role in integrity of the gram-negative cell. This is a protein called bronze lipoprotein, shown in blue here. And notice that it has the purpose of being like a safety pin. It pins together the outer membrane and the thin peptidoglycan layer. So notice that in a gram-negative cell, the most outer exposed layer is the outer membrane. Now, the outer membrane is really different than the cytoplasmic membrane. Sometimes we call the cytoplasmic membrane the inner membrane, and we say that a gram-negative cell has two membranes, an inner and an outer membrane. The cytoplasmic membrane, remember we talked about it in lecture as being kind of like the bouncer at a dance club. It only lets in things with low molecular weight and those things that are hydrophobic. However, the outer membrane is much less exclusive. It lets in a lot of stuff, and that's partially made possible by the porin proteins. The porin proteins let in things like disaccharides, monosaccharides, amino acids, and other forms of nutrients. They won't allow really huge things, but they allow a certain amount of flow. Notice that the outer membrane has, as every good membrane does, an inner and an outer leaflet. So the inner leaflet here is made up all of phospholipids, whereas the outer leaflet is comprised primarily of another molecule called lipopolysaccharide. Lipopolysaccharide, just as it sounds, is lipid and carb. Lipopolysaccharide. There's a long carbohydrate chain that sort of flaps in the wind there and makes the outer membrane of a gram-negative really kind of slippery and mucilaginous. In fact, one of the roles of lipopolysaccharide is in allowing gram-negative cells to adhere to surfaces and form complex slime-encased communities called biofilms. How rad is that? To be able to form a community, maybe a community that adheres to a surface. Ooh, in the case of Christian Graham's work, maybe a community of bacterial cells that causes infection and adheres to the surfaces of the lung, forming a biofilm within the lung. Lipopolysaccharide also has some other roles. It actually is antigenic. Antigenic, meaning that it causes your body to say, foreign invader. That is, it triggers your immune reaction. And in fact, there's a region called the lipid A region, which is the region that's down here in the membrane. And the lipid A region is toxic. Together with the O side chain, this triggers your body to react and to mount an immune response. So LPS is a very important part of the outer membrane of a gram-negative cell. The outer membrane is very protective. And in fact, the outer membrane of a gram-negative prevents certain things such as antibiotics from getting to a gram-negative cell. However, interestingly, recognize that this is the kind of cell that loses the crystal violet dye. So let's talk about the gram stain mechanism, and then we can think about why it might be that the gram-negative cell loses the crystal violet dye, whereas the gram-positive cell holds tight to that crystal violet dye. So when you perform a gram stain, it is going to utilize more than one dye. In fact, two. That's why it's a differential stain. It's going to use a pretty pink safranin and a pretty purple crystal violet. Now, we're going to go through this gram stain step by step, but before we do that, I want to remind you to go to the virtual edge and be certain to watch the procedural elements of this gram stain. Make sure you go to Lecture 3, Experiment 5, and find your way to the gram stain procedural video and review. This is a fantastic way to get yourself ready for the procedure. I want to go through the theory of this, and we're going to zoom on in after you've prepared your awesome smear, remembering the nuances of smear preparation. You're going to have your bacteria all on one slide. Now, your bacteria may be a mixed culture. You may have some rods and you may have some cocci. I'm actually going to look at a mixed smear where we have rods, all rods, and we want to differentiate those that might be gram-positive from those that might be gram-negative. Now, in your notes, it shows coccus and a rod. I wanted to use this to make the point that you can have gram-positive rods and gram-negative rods, and you can have gram-positive cocci, and you can have gram-negative cocci, but understand that it's the color that they stain that tells you what their gram reaction is. Now, it is true that more often cocci that we work with in the lab are gram-positive, whereas we don't work with gram-negative cocci. However, it's important to understand that it's the stain that tells you whether or not an organism is gram-positive or gram-negative. So we begin off with a smear with transparent cells. Everything is transparent. We then hit our smear with crystal violet. We allow the crystal violet to stain all of the cells. Remember that it is a cationic stain or a direct stain, a positively charged stain that directly coats the negative charges of both the gram-positive and the gram-negative cell. Gram-negative cells get their negative charge from the lipopolysaccharide, just as gram-positives get their negative charge from the teichoic and lipoteichoic acids. So everything turns purple. The entire smear is purple. And we want to enhance that purpleness. So in order to get everything to hold on to the crystal violet, gram-positive and gram-negative cells, we add what we call a mordant. That is Gram's iodine. So after adding the crystal violet, which I've got right here, that's the first step. Then you're going to add this iodine. Now, I don't know if any of you have ever worn mascara, but I kind of think about iodine as being like mascara. It enhances your eyelashes. Just in the same way, iodine enhances the staining of the crystal violet. In chemical speak, it increases the size of the complex iodine binds to crystal violet, making it more likely that the gram-positive cell will retain that complex. At this point, even after the iodine, everything is purple. We now will decolorize with ethanol. This is the most artsy step. Please watch the virtual edge to see why. You'll decolorize until the runoff is uncolored with ethanol. Please be certain to use ethanol in the yellow-topped squirt bottles rather than acid alcohol. This is the important ethanol that you need to use for the gram stain. Now what happens at this point, this is the pivotal step of the gram stain. It's everything. Because remember, the gram-positive hold on. They hold tight to the crystal violet iodine complex, whereas the gram-negative cells allow that to slip out. Now here we see that the gram-negative cells have become clear, whereas the gram-positive cells have remained purple. But of course, it's very difficult to see a clear cell under the microscope. And so in order to visualize our gram-negative cells that have lost the crystal violet dye, we add our counter stain. Our counter stain is safranin. It's a beautiful pink color, kind of like the color I have on my lab coat right here. So we'll see it turn the gram-negative cells to a beautiful pink. It too is a cationic direct positive stain. So it's going to coat the negatively charged surface of the gram-negative cells, staining them pink. So we will now have purple gram-positives and pink gram-negatives. And that's going to allow us to differentiate gram-positive from gram-negative. The stain that you're going to be performing in lab is one in which your gram-negative cells are Escherichia coli, E. coli. Gram-negatives tend to hang out in our gut. Gram-positives tend to hang out on our skin and in our nasopharyngeal area. So things like E. coli are gut bacteria, whereas things like Staphylococcus epidermidis, that epidermis, right, is our skin cells, that is something that's a normal flora of our skin. So you're going to be coloring the gram-positive Staphylococcus purple because it's gram-positive, whereas the rods, the E. coli, are going to be gram-negative. So both bacteria, they're both bacterial cells, but one is gram-positive, one is gram-negative, and they stain different colors and come up with a really pretty beautiful stain. I want to show you some models that my students have made in the past. The first one is a model that'll help you remember the order of events in the gram stain. This model, they used spools and wood to make this and then painted everything. Come in and stain. Crystal violet, iodine, alcohol, safranin. Notice when crystal violet is utilized, everything turns purple. Iodine enhances that purple color. When alcohol is run over the gram-negative cell, it loses everything. How embarrassing, right? It becomes completely naked. And then when it is counterstained with the safranin, it turns pink. Purple gram-positives, pink gram-negatives. Come in and stain crystal violet, iodine, alcohol, safranin, and that'll help you remember the gram stain forever. I also have some wicked cool models that students have built of what a gram-positive cell wall looks like versus a gram-negative. This one's pretty artistic. I think I might take it home and hang it up on my wall at home. That you can see this is a gram-positive, right? Thick peptidoglycan, teichoic and lipoteichoic acids. Here's her rendition of a gram-negative. This is Lily. She's got her inner membrane, cytoplasmic membrane, thin peptidoglycan, bronze lipoprotein, and the outer membrane. And she's trying to differentiate the lipopolysaccharide there. You can see it's got the carb region up there. Now, here's one of my favorite models. I'm sad that it's falling apart because it shows the thick peptidoglycan layer in a very three-dimensional way. So you can see that very thick peptidoglycan layer, the teichoic and lipoteichoic acids. This is a beautiful gram-positive model. I've got several others, and these will be hanging around in both lab and lecture. So gram-positive, thick peptidoglycan, lipoteichoic, right, attached to the membrane. Here's our gram-negative. See if you can pick out the porin proteins and the lipoproteins and the thin peptidoglycan on there. But remember that the question we were left with was, why is it that this gram-positive cell hold on to the crystal violet iodine complex, where the gram-negative cell simply allows that complex to escape when treated with ethanol? It has to have to do with the thick peptidoglycan layer versus the high lipid content of the gram-negative cell. But it's fascinating that it was over a century after Christian Graham that we began to understand the mechanism of the gram stain. And in fact, there's a publication that I want to show you of the gram stain mechanism that's entitled "The Gram Stain After More Than a Century." And in the abstract of this publication, we can read, "The gram stain, the most important stain in microbiology, was described more than a century ago. Only within the past decade, however, have we begun to understand its mechanism." We understand that, that the cell wall of gram-positive microorganisms is responsible for retention of the crystal violet iodine complex. In gram-negative cells, the staining procedures damage the cell surface, extracting those lipids, resulting in the loss of dye complexes. So it is the high lipid content of the gram-negative cell that allows the alcohol to damage the cell and allow the escape of the crystal violet and iodine complex. So setting free the crystal violet iodine complex, a gram-negative cell becomes transparent after treatment with alcohol. So that tells you that in order to get an accurate gram stain on any of your cells, your unknown included, which you're going to be gram staining today, you need to make sure that you have fresh cultures, because if the integrity of the cell wall is damaged, then of course the staining capacity of that cell wall won't be retained. Let's turn our attention now to talk about the last differential stain for today. This is the acid-fast stain. This is very different than the gram stain. In an acid-fast stain, we're looking at the ability of a cell to hold on to a primary stain even when we hit it with acid. That is, we say that these cells are acid-fast. The term fast has two definitions. One is obviously going really fast. The other is to hold on to or to hold tight to. It's that latter definition that we're using here. A cell that is acid-fast holds on to the primary stain even after being treated with acid alcohol. The characteristic of an acid-fast cell that allows it to do that is that it has a high content of this waxy hydroxylipid called mycolic acid. Mycolic acid is it's a very waxy layer that's a kind of outer layer of the acid-fast cell, and I'm going to show you a picture in a minute, that allows the cell to hold on to the initial stain, which is called carbylfusion, a carcinogenic stain, by the way. It holds on to that stain even when treated with acid. Now, this stain actually turns out to be diagnostic because those bacteria that cause tuberculosis and leprosy are actually acid-fast. Those two bacteria are both mycobacterium. Now, look at this term, mycolic acid. Mycobacterium. See, the first four letters in both words are the same. Mycobacterium have mycolic acid. I hope that helps you remember for future. So mycolic acid characterizes these pathogens. So if someone is coughing up bloody sputum, maybe you've seen Doc Holliday or you've seen Nicole Kidman in Moulin Rouge when they die of what used to be called consumption, which is tuberculosis. And if you take the sputum of someone who's suffering from tuberculosis, you will see that it will contain acid-fast bacteria. And if you see those acid-fast bacteria, you can diagnose the case of tuberculosis. Leprosy is a disease that often is associated with a more historical context, though there are still individuals that suffer from leprosy. It is a disease that is often contracted in childhood and then doesn't manifest until adulthood. That's because of the very slow doubling time of Mycobacterium leprae, 33 days. That is, you can see that it takes years and years before you actually begin to see infection with Mycobacterium leprae. This, too, is a stain that you want to perform on fresh cultures because it relies upon the integrity of the cell wall. And here's what that cell wall looks like. Kind of reminiscent of a gram-positive cell in that it has a thick layer of polysaccharide. However, it has only a thin layer of peptidoglycan and then a layer of something called a rabinogalactan, another polysaccharide. However, what really distinguishes an acid-fast cell is its mycolic acid. The mycolic acid is the thing that allows that cell to retain the carbylfusion dye even when treated with acid alcohol. Now, I don't have any models of an acid-fast cell wall. Maybe that's what you'll want to look to do for extra credit. It's a pretty sweet cellular structure. Let's look at the staining procedure. Once again, we will begin with a primary stain. This time, though, the primary stain that we use is carbylfusion. This is a phenolic compound, and due to that nature, it is carcinogenic. Thus, when you proceed to the hood to perform the stain, we ask that you wear gloves. You'll be using dry heat to drive this into the waxy mycolic acid. You're going to want to make sure you have a thick enough smear so that you can get enough bacteria to take on the dye and to adhere to the slide. Because you know how wax behaves in water? Wax doesn't particularly like to adhere to things. It kind of chunks up and clumps up, and that's what these bacteria are going to want to do too. So be sure to mix it really well in your water and then dry heat it so that we kind of melt it a little and get the carbylfusion in. All of the cells are reddish pink at that point with this beautiful carbylfusion dye. We're then going to decolorize, rather than with ethanol, with acid. So make sure you grab the little bottle for decolorization called acid alcohol. This is a hydrochloric acid mixed into ethanol. So be certain to recognize the difference between this and regular alcohol. This is your differential step because, of course, those bacteria that are mycobacterium, they're going to hold fast to the carbofusion, whereas those bacteria that are non-acid fast are going to let it go. Then we're going to have clear non-acid fast cells and pink acid fast cells. But it's tough to see those clear cells once again, so we're going to stain them with a counter stain, methylene blue. You've met it before when we did the direct stain. It's a direct stain. It's going to coat the surface of those non-acid fast cells and leave us with blue non-acid fast cells and really pretty pink acid fast cells. Now, remember, the difficulty of this stain is that mycobacteria like to clump together. And in our lab today, we're going to be utilizing Staphylococcus epidermidis as our non-acid fast cell. So your non-acid fast cells will be circles. But remember that you could have non-acid fast cells that could be rods as well. And that's why I use this slide to demonstrate that. In your notes, you'll notice that your non-acid fast cells are cocci. So we can have both cocci that are non-acid fast and rods that are non-acid fast. However, Mycobacterium, our acid-fast bacteria, are going to be rods, although they're pleomorphic. That's the rad thing about them. Remember, pleomorphic means shape shifter. So depending upon the age of the culture, they'll kind of round over sometimes and be more coccus looking. But in our cultures, they should be fresh. And so we should see them as rods, these beautiful pink rods. And if you did a good job of really mixing your smear, you might see individual rods. Otherwise, you'll see big clumps of pink where the mycobacteria have all clumped together. And then your non-acid fast cocci that are the Staphylococcus epidermidis that you stain, non-acid fast, they let go of that acid fast primary stain. So today we will perform 2 differential stains. Get jazzed, get ready, make sure you watch Virtual Edge, and be ready to rock and roll when you hit the lab door.